Hello, in this video we're going to look at cellular respiration and photosynthesis. So what is cellular respiration? Cellular respiration is a bunch of chemical reactions that convert glucose into ATP. Cellular respiration takes place in the mitochondria, except for glycolysis, which takes place in the cytoplasm. The formula for cellular respiration is glucose and oxygen are converted into carbon dioxide, water, and energy. Cellular respiration is broken down into four stages: glycolysis, acetyl-CoA synthesis, citric acid cycle, and oxidative phosphorylation. The first stage, glycolysis, is when glucose is converted into pyruvate. The second stage, acetyl-CoA synthesis, is when pyruvate is converted into acetyl-CoA. The third stage, citric acid cycle, is when acetyl-CoA is broken down into electron carriers and ATP. The fourth and last stage, oxidative phosphorylation, is when electrons are donated to create ATP. Glycolysis, also known as the splitting of sugar, is split into three subphases. The first phase is when you add two ATP molecules to glucose to produce fructose 1,6-bisphosphate. The second stage is when fructose 1,6-bisphosphate is converted into two molecules of glyceraldehyde 3-phosphate. The final stage is when the two molecules of glyceraldehyde 3-phosphate molecules are converted into pyruvate by reducing NADH plus to NADH and adding phosphates to ADP molecules. The next stage of cellular respiration is acetyl-CoA synthesis. This is where pyruvate is converted into acetyl-CoA when NAD plus is reduced into NADH and carbon dioxide is created as a byproduct. The third stage of cellular respiration, the citric acid cycle, also known as the Krebs cycle, is when acetyl-CoA is converted into CO2 electron carriers NADH and FADH2 and ATP are produced. The first step of the citric acid cycle is when oxoacetate is combined with acetyl-CoA to produce citrate. The second step is when H2O is removed from citrate to produce cis-aconitate. The third step is when cis-aconitate is turned into alpha-ketoglutarate when CO2 is removed and NAD plus is reduced into NADH. The same thing happens in the fourth step. NADH is produced and CO2 is removed to produce succinyl-CoA. The next step is when succinyl-CoA is converted into succinate when a phosphate is added to ADP to make ATP. In the sixth step, succinate is converted into fumarate when FAD is reduced into FADH2. In the next step, H2O is added to fumarate and fumarate becomes malate. The final step is when malate becomes oxaloacetate when NAD plus is reduced into NADH and the process repeats again. The final step of cellular respiration is electron transport chain. This is where the electron carriers NADH and FADH2 pump protons and transfer electrons. In complex 1, NADH provides an electron and a hydrogen proton is pumped through. The electron is transferred to complex 2 by coenzyme Q and cytochrome C, where FADH2 becomes FADH and then another electron is added and a proton is pumped. The electrons continue through complex 3, where a hydrogen proton is pumped with the energy of the electrons. In complex 4, oxygen grabs the electrons along with the hydrogen to become H2O, and another H plus is pumped through. All of the hydrogen protons move from high concentration to low concentration through the ATP synthase, providing ATP synthase with the energy to add a phosphate to ADP to produce an ATP molecule. Cellular respiration also has the fermentation process, where pyruvate is converted into lactic acid or ethanol. Lactic acid fermentation is when NADH is converted back to NAD plus and lactic acid is formed. Ethanol fermentation is when CO2 leaves pyruvate to create acetaldehyde and NADH is converted back to NAD plus to produce ethanol. Most of the ATP in cellular respiration is produced at the electron transport chain. By the end, 32 ATP molecules are produced. Cellular respiration is regulated by the molecules ATP, ADP, NAD+, and NADH. For example, in glycolysis, ATP either activates or inhibits the process. When ATP levels are low, glycolysis is activated by PFK1. When ATP levels are high, the same enzyme PFK1 inhibits or slows down glycolysis. Now, let's move on to photosynthesis. What is photosynthesis? Photosynthesis is making glucose and oxygen from carbon dioxide and water. Photosynthesis is a redox reaction where CO2 is reduced into glucose and H2O is oxidized into oxygen. Photosynthesis has two parts, which are the photosynthetic electron transport chain and the Calvin cycle. The photosynthetic electron transport chain is when electrons are transferred and protons are pumped to create ATP via the ATP synthase. The photosynthetic electron transport chain is embedded in the cycloid membrane and all of the reactions take place in the lumen. When light hits a reaction center of a photosystem, it excites an electron. The excited electron in photosystem 2 moves to cytochrome B6F complex passing through the plastoquinone, and two water molecules are split to replace the missing electron in the photosystem 2. The cytochrome B6F complex pumps hydrogen protons with the energy of the excited electron. When light hits the reaction center of photosystem 1, an excited electron is passed to ferredoxin. When ferredoxin passes the excited electron to NADP reductase, NADP reductase reduces NADP plus to NADPH. Electrons lost by photosystem 1 are replaced by electrons from photosystem 2. Just like cellular respiration, ATP synthase pumps hydrogen protons from high concentration to low concentration, causing it to get the energy to add a phosphate to ADP to make ATP. The energy of the excited electrons is shown in the Z scheme. Sometimes when light levels are high, energized electrons are transferred from ferredoxin back to plastoquinone for the process to repeat. Sometimes the energized electrons reduce into O2 instead of NAD+, which causes reactive oxygen species. Reactant oxygen species are converted into H2O when antioxidants are added. The next step of photosynthesis is the Calvin cycle, which is split up into three parts: carboxylation, reduction, and regeneration. In carboxylation, Rubisco catalyzes the reaction between CO2 and the 5-carbon molecule Rubisco 1,5-bisphosphate. The result is two molecules of 3-phosphoglycerate, which are then reduced into triose phosphate with ATP turning into ADP and NADPH turning into NADP+. ATP is required for the regeneration of rubisco-1 5-bisphosphate from triose phosphate. The process of photorespiration is just like the Calvin cycle, except O2 is added instead of CO2, and this concludes photosynthesis. I hope you learned more about cellular respiration and photosynthesis in this video.